Hello guys, this is Geethanjali. In the previous video, we started with principle of inheritance, and in this video, we'll be doing inheritance of one gene. So first, let's dive into the convention that we use to represent alleles. So we usually use a single letter to represent an allele. So let us take an example of a character, height. Okay, so height, either the plant could be tall or dwarf, right? So since here the dominant trait here is tallness, so we will use T as the letter, and if it's capital T, that means it represents the dominant allele, whereas if it's a small t, it represents the recessive allele. So these two alleles together will be called a genotype. So genotype is the genetic composition. Okay, genotype is something that you cannot determine just by looking at the plant. Suppose you have a tall plant, this can either be capital T or capital T small t. So we cannot determine just by looking at the plant if it has both dominant alleles or either one of them as dominant alleles, because when there are two different alleles, such as in this case, the dominant one expresses itself over the other. So this genotype will lead to a phenotype, okay, which is a tall plant. So phenotype is nothing but the character, okay. So when I say a plant is tall, that is basically its phenotype. Now, coming to the other genotypes that we may see, so we can see same type of alleles too. Both can be dominant, or we can see same types of alleles in which both are recessive. Okay, so all these are genotypes itself, and these genotypes have different terminologies for them. So here you can see two different types of alleles, so they are called heterozygous. Okay, whereas these two genotypes, we call them homozygous because they have same type of alleles, right? Both have same types of alleles in them. Now, this is called heterozygous dominant because we know that in different pair of allele, always there is one which dominates the other. Here, the first one is called homozygous dominant, whereas the second one is called homozygous recessive. Okay, so in both the cases, you will find the plant tall, whereas in the last case, you will see a short plant. Okay, so basically, this is the convention that is involved. Now, what is Punnett square? Okay, so Punnett square was given by Renegade C. Punnett. Okay, so Renegade C. Punnett devised this Punnett square, and it's very helpful to understand many crosses and all that. So this is nothing but a type of table or a graphical representation, which we can say, to find the probability of all types of genotypes, all possible genotypes you can see in the offsprings when you have a genetic cross. Okay, so let us see what experiment did Mendel do. So first he took a tall plant, a true breeding variety. Okay, what do I mean by true breeding variety? True breeding variety means it is homozygous. Okay, both are homozygous. That means they have capital T, capital T, capital T, small t, small t. Okay, so when he crossed them, he found that all the plants that he got in the first generation were tall. Okay, without any exception. So this first generation, this hybrid generation is It's called filial first generation, okay, and this is a filial second generation. So filial second generation was obtained by again mating among the F1 generation. So when this happened, they found out that 3 out of 4 plants were tall and 1 out of 4 plant was short. Okay, so to explain this, we need to look into this diagram. Okay, so we know that when gametes are produced, okay, they take half of the chromosomes with them, right? They only have half of the chromosomes with them. So they will either have one of the two alleles. So we have two alleles in general. So when they take the chromosomes, they carry half of the chromosomes. So the gametes will take either one of the allele, right? So here you see that when there is a tall plant, homozygous tall plant, you will obtain only one type of gamete which contains capital T allele. Okay, whereas here also you will find that it contains only one type of allele. All the gametes are only of one type, that is, they will contain only the small t gene, the small t allele. Now, if you put this across a Punnett square, you will get heterozygous everywhere. Okay, so this heterozygous, we know that this is always dominant. Okay, so we know that heterozygous will always be dominant, and the dominant character that you see here will be the tallness. Okay, we will see all of them are tall plants. Now, if you cross among them again, see, these all are again tall and there is no short plant here. But when you cross these tall plants, you will find out that here we saw that when we cross tall plants, we are getting one dwarf over here. So that was quite a bit surprising if you don't know the reason behind it. So when he worked out the reason behind it using all this mathematical statistics, he basically found out that, so in this case, now there will be two types of gametes produced, not one type as in the previous case. So one will be carrying the dominant allele, other will be carrying the recessive allele. So both the plants will be producing two types of gametes, and since they are producing two types of gametes, there will be four possible combinations. Okay, and out of this four possible combinations, you will find out that two of them are same. Okay, and the other two are different. So in this case, you will find all types of genotypes here. Okay, so you will find heterozygous dominant, you'll sorry heterozygous dominant, homozygous dominant, and homozygous recessive. Okay, so this is the ratio, 1 is to 2 is to 1. So you will see you have one homozygous dominant, one homozygous recessive, and two heterozygous dominant. So the genotypic ratio for the F2 generation is 1 is to 2 is to 1. Whereas the phenotypic ratio, the phenotype means just the character. So here when we say phenotype, we will only consider tall tall plants. Okay, so we can see there are three tall plants and there is one short plant. So in that case, we'll say the phenotype ratio is 3 is to 1. In this entire cross, we see that we only took one character. Okay, we considered only one character, that is this height of the plant. So that is why this is called mono hybrid cross. Now, how did exactly Mendel find out that these were some of them were heterozygous and some of them were homozygous in these tall plants. So for that we need another cross, that is test cross. So it is called test cross because we are basically testing the genotype of the dominant character of the plant. So in this cross, what we do basically is we take, let us say, one of these plants. Okay, we don't know if it's a homozygous or heterozygous. So we take one of them. Okay, it can either be homozygous. Here they have taken the color, okay, instead of the height. So the color, either it can be violet, okay, or either it can be white. Either it can be white or violet. Violet is the dominant trait, okay. So in this case, either it can be homozygous or heterozygous, right? So we have to find out that. To find that, we basically cross one of these with the homozygous recessive parent. Okay, so you can see homozygous recessive parent, that is the one with the white flowers, is crossed with the unknown with the unknown flower. Okay, so then let us say if it was homozygous, then we know that when homozygous dominant and homozygous recessive they breed, we have seen here itself in this case that all of them will be giving the dominant character. So when you find all of them giving you violet colors, you will say that this was a homozygous plant that we used. But in case you find a difference in that, let us say you find 50% I mean dominant and 50% recessive character, then you'll say that the plant that we took was heterozygous in nature. Because see, according to this Punnett square, we can see that we'll find that half of them are heterozygous dominant and other half are homozygous recessive. So in this case, the plant that we took will be heterozygous. So this was test cross and monohybrid cross.